Good day, everyone. Today, we'll be presenting our study entitled From Molecule to Medicine Computational Development of Kanglemycin A Against Multidrug-Resistant Tuberculosis. Our study focuses on developing a potential treatment for MDR-TB by combining computational drug design with a targeted pulmonary drug delivery system. Rather than stopping at identifying a promised drug, we also designed a formulation that could deliver it more effectively to the site of infection. To start, MDR-TB is a form of tuberculosis that is resistant to rifampicin and isoniazid, the two main first-line anti-TB drugs. Because of this resistance, treatment becomes much longer, more complicated, and often less successful than treating drug-sensitive TB. This highlights the need not only for new antibiotics, but also for delivery systems that can improve how these drugs reach the infected lungs. Now, our first step was selecting the therapeutic target and the drug candidate. We chose the beta subunit of RNA polymerase, which is a protein essential for bacterial RNA synthesis. Since the bacteria cannot survive without producing RNA, inhibiting this protein prevents bacterial growth. However, mutations in RPOB are also the main reason why rifampicin becomes ineffective in MDR-TB. To address this, we selected Kanglemycin A because it is structurally related to rifampicin but contains additional functional groups that allow it to bind more effectively to certain mutated forms of RPOB. This makes it a promising candidate for overcoming rifampicin resistance. After selecting the drug candidate, we determined the most appropriate route of administration. Oral delivery is simple, but only a portion of the drug reaches the lungs because it must first be absorbed and distributed throughout the body. Intravenous delivery provides rapid systemic exposure, but is invasive and may increase unwanted side effects. Since tuberculosis primarily affects the lungs, we selected a pegylated nanoliposomal dry powder inhaler. By delivering the drug directly to the lungs, we can achieve higher local drug concentrations at the site of infection while reducing unnecessary systemic exposure. Next, we evaluated the pharmaceutical properties of Kanglemycin A. Its poor oral bioavailability makes pulmonary delivery a more suitable option than oral administration. In addition, its relatively short systemic half-life supports the need for a biphasic release profile, while its activity against RPOB makes localized delivery to infected lung cells a logical strategy. Overall, these characteristics support the use of a nanoliposomal dry powder inhaler. Based on these properties, we designed a biphasic release profile. The first phase provides rapid drug release immediately after inhalation, allowing the drug to begin inhibiting bacterial RNA synthesis as early as possible. The second phase then releases the remaining drug gradually over the next 12 to 24 hours, helping maintain therapeutic drug levels in the lungs. We selected this release profile because it combines the advantage of a fast initial effect with prolonged drug availability. As a result, it may reduce dosing frequency, improve patient compliance, and minimize systemic toxicity. Finally, we selected the components of our formulation. Kanglemycin A serves as the active pharmaceutical ingredient. HSPC and cholesterol form the nanoliposomal membrane and help improve its structural stability by reducing premature drug leakage. DSPE-PEG 2000 provides a protective PEG layer that prolongs the residence time of the liposomes in the lungs before they are cleared. Trehalose protects the liposomes during spray drying, while lactose monohydrate serves as the powder carrier and L-leucine improves powder dispersion, allowing more particles to reach the deep lungs. Together, these components create a stable formulation capable of targeted pulmonary delivery and controlled biphasic drug release.
Kanglemycin A has a short 2.4-hour half-time, limiting its MDR-TB efficacy. Our solution is a rehydrated nanoliposomal carrier that maintains therapeutic levels for over 24 hours. This maximizes local lung action while minimizing systematic side effects. We engineered particles at 1 to 5 microns to ensure deep lung deposition and avoid airway trapping. The PGLA layer ensures sustained release while IDSPE PEG 2000 stat shield delays macrophage clearance. Finally, cholesterol and HSPC optimize membrane stability. We utilize a biphasic release profile to solve the drug's short half-life. Phase 1 provides an immediate burst release within 1 to 5 minutes to rapidly halt MDR-TB replication. Phase 2 ensures sustained diffusion over 12 to 24 hours as the polymer backbone brigade is providing constant localized efficacy from a single dose. We control formulation via three knobs. First is bilayer rigidity, prevents premature drug leakage. Second is surface steric hindrance, creates a stealth shield against immune detection. And third is powder dispersibility, lowers surface energy to ensure efficient deep lung delivery. We use a clinical DPI device to deliver our engineered powder. By coating lactose carriers with L-leucine, we reduce surface energy and static adhesion. This design effectively bypasses upper airway trapping, ensuring the medication consistently reaches the target alveolar region. This is the visualization of utilizing the dry powder inhaler with the crystals into the lungs. Manufacturing uses two stages: nanoprecipitation. Microfluidics enable precise, spontaneous self-assembly of liposomes. Second is spray drying. Atomization through a two-fluid nozzle creates highly uniform, free-flowing porous particles at the ideal 1 to 5 micron size. Our validation confirms success. In aerodynamics, 78.5 fine particle fraction or FPF proves deep lung access. Encapsulation of 92.1 efficiency shows high drug retention, and efficacy in silico modeling shows Kanglemycin A binds 2 times stronger to the mutated target than standard treatments. Step 10 is our quality safeguard. If targets aren't met, we evaluate. Adjust lipid or PEG ratios, recalibrate spray drying parameters, and retest until we consistently hit our aerodynamic and pharmacokinetic benchmarks.